once we have our concentrated pool of proteins, like we have this tube where we have our proteins in, just like we described in the last ones. Now we can actually start to try to break it up and analyze the proteins. Two ways of doing so is through protein hydrolysis and protein cleavage. So in protein hydrolysis, basically what you try to do is you try to cut the chain of proteins into specific amino acids. So if you remember, a protein is just like a long string of amino acids, right? Which are basically, bound together by these things called bonds. Okay. And that means what we want to do is we want to find a way to chop up the amino acids. So we basically only have, we don't have the full protein anymore. We just have the amino acids left. To do this, there are three different methodologies which we can actually use. First is acid hydrology. Hydrolysis. There we go. Now, the opposite, you can also use base hydrolysis. And lastly, also proteases. We're using proteases. So this is called enzymite or enzymate, I think, something like that. Enzymate hydrolysis, okay? So basically, hydrolysis uses water with, with either like acid, base, or enzymes to break open these amino acids, okay? And as the name suggests, acid would be, it would break open this with the acids. Base would be, it would be break these open with base instead, with a high base value. And enzymites would actually use something called proteases, I think that's how you say it, to break open the protein. Now, if you remember actually from the cell separation video, these proteases, they are, like natural proteins which break open other proteins, So we can also use here as an advantage, while during the cell destruction, it would have been actually, something bad. Then once you have your soup of, like, you have here then your soup of amino acids, which are no longer bound to each other. So it's, not a protein anymore. You can use two methods to actually then try to filter them. Similarly, how we filtered and separated the proteins. You can use here ion exchange and also RP-HPLC. Now, these are like chromatographic, solutions again, like we had before, where we had this column, right, with beads. And this time, instead of, trying to attract the proteins to the beads, you would then, using ion exchange, try to attract the amino acids and filter them based on their charge. And you can do that by altering the charge through the salt and pH value of the solution in here. Next, if we look at cleavage, it's actually very similar to hydrolysis, but instead of breaking it into every single amino acids, we actually break it at specific locations, okay? So if you have here again a protein like this, instead of breaking it at every single amino acid, we would say, okay, we would break it at only this place and this place and this place and this place, which is very, very exact. And these, resulting, I guess mini proteins or cut up proteins that you then have, these are called peptides. And this is actually very, very useful for us because we can actually then have not quite a sequence, but we basically can then try to find, based of this, what kind of protein it is. So one way of doing this is through endoproteases. And what we do here basically is we have a protease, like we mentioned before, that cuts every time after lysis or arginine. So let's say this right here is lysis for which the letter is K, and let's say right here this is arginine for which the letter, oh wait, I need to copy it, for which the letter is R. So what would happen basically is we would then cut from here to here, so it cuts at lysis, and then it also cuts at here the arginine. So we then have two pieces basically of this protein. And it does this using the protease, as we mentioned before. You know, these proteins that cut up other proteins, but this one is like very specific. It cuts it at only very specific areas. And then we would take all these peptides which we get and we would try to compare them, you know, with similar proteins through the peptides in a database, in a specific database, and then we can basically view through the peptides what kind of protein this is. Now, there is a different methodology instead of endoproteases where we use chemicals, okay, called chemical cleavage. And we do this with CNBR, which is like a chemical which breaks it always at methionine or something like that. I don't know. I all these biological or chemical terms I'm very, very bad at. So the letter for that is M. So let's say here's M, and then it would cut from here until here, and then from here until here again. And then we basically count the peptides again that result from this, and this is how we then also identify through this methodology what kind of protein this is. And yeah, that has been protein hydrolysis and cleavage. Thank you very much.